Tem que estar azul. Já está azul. Vamos aguardar mais um bocadinho que as pessoas também digam não. É que o workshop era para acabar a 1 e passou um bocadinho. Então as pessoas estão um bocadinho atrasadas, mas não queremos começar muito depois da hora também por causa do
Sim. Eu não vi sim, claro.
Esperar mais um bocadinho que a Madalena refresque alguma coisa que precisamos e depois começamos. Depois se a professora durante as apresentações preferir sentar-se. Ok. Depois vir o computador. Eu vou vir agora. Está a ouvir-nos? Sim. Está a ouvir-nos? Ok. Pronto, então nós vamos agora dar início à sessão. Ok? So, good afternoon, everyone. Thank you for being here with us today. It is with great pleasure that I present you the lecture. I'm sorry. That I present you the lecture, The World of Fungi, A Novel Path in Sustainability, moderated by Professor Cristina Viegas. Here you have the QR code. You can scan it with your camera, so you earn biocoins to win one of our prizes at the end of the week. Let me remind you to submit all your questions through the corresponding section of the SBE app. By doing this, you will also earn biocoins, and stay tuned until the end of the lecture because there is a giveaway with a small prize. So I hope you enjoy this lecture. You find it as interesting as we hope. And without further ado, I hand the floor to Professor Cristina Viegas. Thank you.
Okay, thank you. Good afternoon. First of all, I want to thank you for the invitation to share this session on the world of fungi. So fungi are well known for their important roles in nature, in ecosystems, and also in so-called traditional biotechnology. So since a long time ago, mostly as providers of food products since ancient times, when the presence and the roles of fungi were not yet known, and they were still already there working for us in some way, for the humans. Some of the fungi can otherwise be detrimental in relation to disease, to food biodeterioration, to materials biodeterioration, etc. And as we know, the risks associated with these aspects are predicted to increase due to climate changes. Some alterations are already on the way. Besides these more traditional aspects, many fungi play pivotal roles in modern biotechnology process. And for sure, our speakers will bring us today very interesting novelties about these amazing microorganisms, and mostly, as far as I know related with these more modern aspects of biotechnology. So our first speaker is Doutora Maria da Luz Calado. She holds a PhD in biology, in the specialization in microbiology by the Faculdade de Ciências da Universidade de Lisboa. She is presently, as far as I know, in the researcher in the MAR, Centro de Ciências do Mar e do Ambiente, and her work is focused on the study of the biotechnological potential of marine fungi from the Portuguese coast. At least I think this is one of the aspects of her work, and she will give us a more complete story about her work. So I give the word to Dr. Maria da Luz Calado.
Thank you so much.
She can't be, I'm sorry, she can't be here for personal reasons, but she's in the Zoom and she will make her presentation in the Zoom.
Can you hear me?
Yes.
Yes.
Yes? Okay. First of all, good afternoon. And also I would like to thank you, the organizing committee for this event, for this invitation, and for having the opportunity to share my work, particularly the work that I'm developing in the last few years. And second, I would like to apologize for not being there for this situation, but it was a situation that I was not able to solve. So, well, we have, fortunately, we have this way to communicate, and it's great. So, I'm going to share my, let's see if I can do this. Am I sharing? Or not?
Not yet.
Now? And now?
Não,
No?
já deram,
Oh, my.
deram permissão para...
Eu creio que sim.
Let's see.
Ok, agora
I
parece
know.
que já aí vem.
Okay. My mistake, I'm sorry. Okay, so my lecture will be about marine fungi. That is the topic of my research, and the question that I want to solve is, well, that I'm going to talk is, why should we focus on these particular microorganisms? So marine fungi present an ecological group of fungi that occur in marine environments, from intertidal habitats such as mangroves, salt marshes, or the beaches, and they go to deep oceanic habitats. In these habitats, they might colonize a different range of marine substrates depending on their nutritional requirements and strategies. So they might be saprobic, parasitic, or symbiotic. They might have this nutritional lifestyle, and also they might occur as endophytes. In that case, they are living in the internal tissues of their hosts. That in these environments might be seagrasses, elophytes, or seaweeds, and living in these tissues, apparently they are not causing any disease symptoms to their hosts. So the definition, the concept of marine fungi has been recently updated. So according to this definition, the group of marine fungi includes any fungus that is recovered repeatedly from marine habitats because it is able to grow or sporulate on substrata in marine environments. It forms symbiotic relationships with other marine organisms, or it shows to adapt and evolve at the genetic level or be metabolically active in marine environments. So this means that we might consider marine fungi all the species that are exclusively associated with marine ecosystems. In that case, we are talking about marine fungi sensu stricto, but also species that demonstrated to be physiologically well adapted and metabolically and functionally active in these ecosystems. In that case, they are called osmotolerant or allotolerant fungi. In this last group, we include a few genera that we all know. They are well known to occur in terrestrial environments, such as Aspergillus, Penicillium, Fusarium, Acremonium, and others. So, in what concerns nutritional strategy, the majority of marine fungi sensu stricto is composed of substrates of plant, seaweed, and marine origin. And osmotolerant and allotolerant fungi have been mostly reported as endobionts of intertidal halophytes, seagrasses, seaweeds, and animals. But they also might occur as decomposers of organic matter retained on the marine sediment matrices. But why are they important? Why am I talking about marine fungi? Because recently, well, not that recently, but we have lots of papers published about this, but they have been recognized as an emerging and prolific and promising source for the production of chemically new and structurally unique secondary metabolites. And these metabolites have been tested and show it at the moment very interesting bioactivities such as antimicrobial, antiviral, antitumoral, antioxidative, anti-inflammatory, photoprotective, among other properties. And also, they might produce extracellular hydrolytic enzymes that might be used in that might have biotechnological applications. So, but why are these natural products that are produced by these fungi different from those produced by terrestrial strains? Now, the answer is we don't know yet, but most of the studies pointed out suggested that there are differences. And these differences seem to be related with the fact that these fungi are exposed to very variable and harsh environmental conditions. And these conditions, and also the fact that some of these fungi are establishing an ecological relation with a host, might also be activating particular biochemical pathways that are responsible for synthesis of some of different compounds. That might be the explanation for this. So we have the fungi that might produce a high number of bioactivities, but also most of these species they grow very easily and rapidly. So the fact that it's really easy to cultivate these species and also to obtain a high yield of secondary metabolites or enzymes turns the use of marine fungi a sustainable alternative to obtain natural compounds instead of the chemical synthesis. So, and which are the major contributors for this? So endobionts fungi that are associated with marine invertebrates and seaweeds stands out among the already tested communities as one of the main producers of bioactive compounds. And also in this group, the fungi that are included in the genera Aspergillus and Penicillus seem to be the most contributors to the production of these natural products. So, considering that we have new compounds that might be safer, eco-friendly, and sustainable, they have multiple applications. They might be used by pharmaceutical industry as natural drug candidates. Also, they might be used in cosmetic industry as photoprotective, anti-aging, skin whitening agents, or they might be used in the food industry as natural pigments, additives, and preservatives. Also in agriculture as biocontrol agents, and many more, such as bioremediation agents, anti-folding agents, and so much more possible applications. However, and despite the increase of studies that focused on these microorganisms, the diversity of marine fungi and the potential natural products synthesized by newer species that have already been reported is not yet represented. And why is that? There are still many chemical underexplored species, and also many more ecological niches and marine environments to be bioprospected. So, given that the main purpose of our research team is to contribute to fill this gap of knowledge. So we have two main goals. First goal is to isolate marine fungi, and including marine fungi sensostrict and also osmotolerant, allotolerant fungi from different hosts or substrates. Then we want to evaluate their potential to produce compounds that have biopotential importance, such as bioactive compounds and extracellular enzymes. So what we have done until now, we started our work in 2018, yes, and we started to collect different seaweeds, different species of seaweeds, the brown, green, and red seaweeds, also sea foam and reed plant substrates from 3 Portuguese western coasts and beaches that are near Peniche, that is near our research center. And then the next step was to isolate the fungi. So we used 2 methods to do that. So the first method was to cut the seaweeds into small fragments and then sterilize the surface to make sure that we are only isolated endophytes. And then we did the direct plating of those segments into the culture medium. We did the same with the sea foam. We did the direct plating. But every time we were able to detect footing structures that the fungi used to reproduce. We detect those footing structures, and then based on the morphology of the footing structures, we identify the fungus. We try to identify the fungus, and then after being identified, we transfer suspension of spores into the culture medium, and then we have this isolate. We have a pure culture of that fungus. So the next step was to identify the isolate. So again, every time we had fruiting structures, we use it to try to identify the fungus based on the morphology of fruiting structures or the morphology of the spores. But most of our isolates were identified based on molecular methods. So we extracted DNA and then we did a PCR and amplified the ITS that we choose as a barcode region. In most cases, with the ITS, we could reach into the genus level, but we had, in most of the cases, choose another gene to reach the identification, to have the identification to the species level. So after the identification, we did a small scale fermentation, which means that we transferred the isolates into the liquid medium that was Wickerham medium, and then let it stand for a variable time period depending on the growth rate. After that, we extract the secondary metabolites that each isolate produced by using ethyl acetate from the fermentation broth and also from the mycelium. In the end, we get the crude extracts from each isolate, and then we screening the biological activities of those extracts. So we screening a lot of biological activities, so, antioxidative activity, and we used 3 methods. Also, antimicrobial activity of the potential antimicrobial activity of the extracts. and we tested against Staphylococcus aureus, Pseudomonas aeruginosa, and Candida albicans. And in some cases, with only a few extracts, we also tested against other microorganisms. We also tested all the extracts that we have to see if they have anti-tumoral activity on the cancer cell lines, specifically human prostate and breast cancer cell lines. And we did other tests. We test the anti-inflammatory, photoprotective, and enzymatic inhibitory activity to some of our extracts. We said these are only some of the analysis that we performed with all the extracts that we have. So at the moment, we are still treating our data because we are a very small team, but almost there. But at the moment, we have around 80 marine fungal species isolated. Most of these species are identified into the species level. And we know that they all belong to the Ascomycota, and they are included, and the taxonomic class is more frequently reported from marine habitats. And for those species that we know the name that we had identified to the species level, we checked and we realized that most of them are being recorded for the first time associated with those hosts or in Portugal. That we performed already, most of the extracts exhibited one or more bioactivities at low concentrations, which suggests a high biotechnological potential. So we have already published a paper, and we have also some posters about these results. But we are hoping that very, very soon we are going to publish more results because they are really, really interesting. And also I want to say that we have another student, we have some students doing their thesis with us, but we have one that is focused only on marine fungi. And the main purpose of his thesis is to isolate the marine fungi associated with sand. So he's intending to isolate or identify the species of the aeroneculus fungi. And also he wants to see if they have an ability to produce several enzymes such as protease, amylase, lipase, and other enzymes. And so far we have really interesting results. So I hope you get so, well, I hope that you enjoy my presentation. I hope to try to, oh my God. I'm trying to get to create this enthusiastic audience just to, if you want to come to our lab to see our research, you are very welcome. We will be really happy to receive you there. Thank you so much for your attention.
Thank you very much for this interesting presentation and for showing us the photos of those the photos of the fungi colonies are always very interesting and very colorful and so on. So it's and the presentation was very interesting and the work. So the discussion will be in the end. So now we are going to have the master Miguel Ramos. Is graduating in microbiology and a master in environmental contamination and toxicology and is on his way to finish his PhD on biotechnology, if I'm not wrong, on the Universidade Católica, na Escola de Biotecnologia da Universidade Católica de Porto, and he has research work with saprophytic and mycorrhizal fungi, and he will bring us another perspective or a different perspective about potential applications of fungi.
I hope. Okay, good afternoon. So, I already been introduced. My name is Miguel Ramos. I come from the School of Biotechnology from Catholic University in Porto, and mainly, specifically in an environmental group. And my field of work is mycorrhizal fungi, more specifically ectomycorrhizal. That is a fancy word for wild mushrooms. But the idea of using this wild mushroom is not only for the typical use is production, food, but is to use this kind of fungi as a biotechnological tool to have some results in terms of symbiotic effects or sometimes other kind of effects that I will show forward. Okay. So nowadays, for those who are not used to be in a culture of mycology, can receive three main perspectives from the mushrooms or from fungi. The first one is the savior side. I don't know if you already saw the Netflix series or documentary that from Paul Stamets that show that fungi can help us do anything, save the world, and even more. But also there is the villain side. That's the Last of Us. That a fungi, in this case a mushroom, has this objective of kill us all. And there is other side that maybe someone knows. That is the fun side, the entertainer side of mushrooms. I think you already know what I'm talking about. Super Mario. I hope it was this kind of mushrooms we were talking about. So, but our perspective is not the savior, the villain, or the fun side. Well, it's a little bit the savior. It's more about the sustainability. That's the topic of our presentation. And so, like I said earlier, we use fungi as a biotechnological tool. And since our main goal is to use mycorrhizal fungi, of course, we have to have these fungi connected to some host. One of our recent works was to apply, in this case, inoculate, another fancy word to applicate the fungi, in this case, to roots of plants in order to help them grow. Our recent work was in urban trees. We had an European project that we had to apply ectomycorrhizal fungi to urban trees that they are used to grow in these small boxes in the middle of the sidewalks on the roads, and they do not have enough nutrients or soil space or even water to survive. And in order to improve the resilience, we have applied these fungi, and we even applied in three main cities. It was in Porto, Strasbourg, and Leuven, so Belgium, France, and Portugal. And the idea was to see in different weathers and different conditions, different soils, if we could have the same effect. Of course, we have to change the fungi, we have to change the techniques, but if we could improve the resilience of the plants. In collaboration with INRA in France, they have connected some sensors to the branches of the tree and the trunk to see if the plant was more healthy. And yes, we could see that was an increase of water and nutrient access. We could boost in terms of diseases, because when a plant is in a tough space, the opportunistic fungi, the other side, the villain side of fungi can appear. It can attack the plants. Or in this case, if we are giving more water and more nutrients, of course, the plant is not that weak and can defend himself. So there is a symbiotic effect, even in terms of defense from other fungi. And the fact that we, depending on the library of fungi we have access, we can choose a different kind of species, but all from ectomycorrhizal area. And we can have this wide adaptation with the same effect between all the spaces, all the cities. In even the size of the boxes were not relevant when we use fungi. Other side is not from typical ectomycorrhizal, is more herbuscular, other kind of mycorrhizal fungi, nowadays is very popular as a biofertilizer. So it uses the same effects, it provokes the same effects that urban trees show, the increase of water, the nutrient, all of that. And so if the plants receive more water and more nutrients, well, the direct consequence is we don't have to use as many synthetic nutrients or fertilizers as we used to. So the fact that we have this all established and is something that is established in terms of agricultural practices, nowadays in terms of Portuguese law, mycorrhizal fungi are already considered a fertilizer, not biofertilizer. Biofertilizer is a European classification, but we already have in this porteria a line that says that if we have a product with herbuscular, or in this case mycorrhiza, we can sell as a fertilizer. Okay, so it's almost not only the fact that academia show the results, or even the agriculture engineering already show the results, is the fact that it's already integrated in our life, in our laws. Okay. Another way to see how fungi can be help us is, and now it's my field of work, it's reforestation. So ectomycorrhizal have this symbiotic association more to forest trees and for forest plantations. So when do we have burn fire or burn forest or have other kind of abiotic stresses, it's useful to have a second partner, or at least a partner, to the tree when we do this kind of transplantation to new fields, because the plant will be all alone in this soil without, well, irrigation, without sometimes nutrients. And the fact that in the first years, the first and the second, it's the most important to establish a plantation. It's very important to have this partner, in this case is a fungi, that we apply in the early days of the life of plants. So we do this mycorrhization in the nursery in order to, the plant when it goes to the land and the soil in the forest will already be connected with the mycorrhiza, with this fungi, in order to help them in the first years. Of course, some of these fungi, they fruit, and some of these fruits can be food. And it's other side of the mycorrhization, of ectomycorrhization of some forest trees, is the fact that in the end we'll have this production of mushrooms, that even so some of them have this economic value that is very important. And since Portugal has a weak perspective about the forest, the fact that it doesn't, well, it grows too slow or doesn't give a lot of financial return, that's a way to change a little bit the angle and the perspectives, because mushrooms will appear in a short period of time. And when we are just counting on the forest to cut the tree, of course, we are thinking about long-term exploration of forest. So this is a way to have some return. And also the fact that as it's connected with social traditions is a way to calling back people to the forest. And when we have this saying in Portugal, when it's away from the site is away from the heart. So if we can bring people back to the forest, I'm sure to the fact that we have to maintain them, clean them, will be not that difficult, because it's connected to other kinds of benefits, and not only about the wood production, or even the fact that we are just creating biodiversity. Okay. This is our main applications of fungi in our environmental group, but there is also other applications. So mycoremediation, it's an area that is full of work with fungi, especially because they produce and they exhale some extracellular enzymes, and these enzymes are very powerful in terms of decomposing big moleculars, especially toxins, and even mushrooms. The fact that this, in terms of typical growth, it grows the mycelium, and this mycelium likes to aggregate and likes to absorb. So even if they can not eliminate, or, well, there is no elimination, but even if you cannot decompose the substance, maybe they can absorb these substances. Is a way to clean land or clean polluted sites. In an amateur side, typically people just collect these pollutants. It can be the typical trials is to use oil, air, gasoline, and impregnate these kind of pollutants to straw. That's the main component for the cultivation of saprophytic mushrooms, and let the fungi to grow. In this case, it will, of course, decompose the straw. That's the main food, the main source of nutrients. But also, when it finish decomposing this main source of nutrients, we'll start decomposing other kind of substances. In this case, oil can be a source of carbon, and then we'll feed on this oil. And so it is a way to treat these polluted matrices. But this is in amateur side. In industrial side, the main use is to growth the fungi to release the enzymes or to extract enzymes and then apply to pollutants. Another way, it's the fact that in marine environments, the fact that there are sometimes these crude spills to sea. One of the main tools they have is to apply enzymes to this crude that is on the surface of the sea, and then to dilute these materials until it's easier to nature to absorb, and then in a way to treat. Or in an exotic way, that's something that we already did in the laboratory. So if the fungi has this main ability to absorb something that it doesn't like, in this case was metal particles, and if it grows around these particles, of course, the fungi, in a way, will become magnetic. So from a stream of a factory that releases iron and other kind of metals, we will be able to grow a fungi that absorb, it's like a filter, absorbs these components, and in the end, we could remove the fungi just applying a magnet. Okay, it's not the typical way to do it, but it works. And last but not least, the fact is very popular these days is the use of fungi to grow materials. It's the right word, is to grow materials. So the typical ones is to create composites that we can see in the upper side and the left side. We can do boards, we can do boxes. Applying the same technique, we can do packaging. It's all come from the same company, it's Ecovative. And we even in our lab try to use the same techniques and the same perspective of growing materials. And we have created the idea was to create a pallet. Okay, pallets use wood and sometimes use nails that when we want to renovate or just to decompose this kind of materials is difficult. So the fact we could use a material that is strong enough as a Euro pallet, but in the end, if you want to eliminate them or decompose them, it can be more friendly. That was our objective. We did Euro pallet, but not the size of Euro pallet. It was even very, very smaller pallet. MicroWorks has nowadays this kind of materials that looks like leather, and they are being used to do shoes, to do even clothing. And nowadays MicroWorks is already selling these, I don't know what to say in English, but it's very big rolls of this kind of material, so to industry to apply them. Okay, so I think in a brief period of time we will see this kind of materials as a part of our clothes. And the last example, it is when I draw the line in terms of sustainability, is when we're trying to create this alternative food for vegans to replace bacon. And when I see this kind of example, even if they use fungi, I ask why. I don't understand why they try to have this alternative, because bacon is good as it is. If you don't like it, don't eat it. But even so, it's an example, and I think you should know there are people using mushroom to try to grow bacon. Hope you like. Thank you.
Okay, thank you very much for more perspectives about the importance and use of fungi. It was very, very interesting. Now I welcome Professor Nelson Lima. He holds a PhD in Engineering Sciences in Biotechnology from Universidade do Minho and is a full professor catedrático in Universidade do Minho. His research has been related to environmental and food mycology. He is the head of a very important infrastructure for the ones who work with and study fungi. That is the Micoteca da Universidade do Minho. That is a very important fungal culture collection in Portugal. And he is responsible for some Horizon European projects related with the culture collection, the MIRI. A thing that I would like to refer is that is related with the importance of Professor Nelson Lima work, is that he gives the name to a fungi species that is a fungi of the genus Penicillium, the Penicillium limae. So it's very
Okay, thank you. Okay, thank you. Good afternoon, everyone. First of all, thank you for your kind invitation to come here to collaborate once again with the bioengineering group and all the group that I know so many years. Today I will not speak focus only in fungi. The two speakers before me, they did a good job. Today I will focus in another different way to see how to collect and how to preserve the biodiversity in ex situ, in collections, and what we can do after this, and how we are working nowadays at European level. Why this is for me is so important? Because we are on the beginning of your careers, maybe some of you scientific careers, and sooner or later we will face the problems that is where I isolate my fungi or my microorganisms, where I deposit the microorganisms, how to preserve them for the future. And the most important is not lost the biological materials that after one thesis or a master thesis or several PhD theses, when we ask, or very nice papers, when we ask about the fungi or the strands that do a lot of things so nice, so beautiful, very well characterized, a lot of money spent on these papers and put on this make science, normally we don't find the microorganisms to continue the work, or even worse, to confirm the results. And science without confirmation of the results is a big problem for us, because you know that the quality of science rely in the peers and the confrontation of the results that each group use and get from the different biological resources. So I will speak about MIRI. MIRI is nowadays a research infrastructure related with microbial resources. That means that are collections. Nowadays, this microbial resource also we can speak, and we enlarge the concept for biobanks. And biobanks, because it is not only the microbial materials or microbiological materials are important, but for human derivative materials like blood or stem cells or organs are also so important to preserve and to keep them on biobanks. So we have similar infrastructures and similar way to work. And from the end of last year, these research infrastructures became a legal entity. So it's completely independent of our national collections. We are a big consortium at the European level, and we became with the label of ERIC, that is a European Research Infrastructure Consortium. That means it's a legal entity. And we work not to concentrate. It's not our idea to put all the microorganisms in one single site. Could be, but it's not our idea. Our idea is to be distributed and to keep the expertise that everyone have in different laboratories with different collections to join us, and what we do at national and European level is coordinate this infrastructure. Why MIRI, Eric? We are an infrastructure, and the infrastructure is to serve the bioscience and the bioindustry mainly. And of course that to try to facilitate the access of microorganisms that we have on the laboratories, on the collections, because so far the different collections, maybe Professor Cristina told about Micoteca da Universidade do Minho, maybe for you is the first time that you're hearing about this. However, there are many collections in Portugal and at the European level and international level. So what is important is to bring all the information, to put the information in one single catalog with similar quality standards, and try to deliver what would we do the best way, that is high quality of microorganisms to facilitate that you can make a good science. And even if you lost the materials for one reason or another reason, we can return all the time to the origin, to the collection, and ask or pay to the strain again. We work under the domain of health and food, agro-food and environmental energy. And of course that for us one key element is to foster the competitiveness at industrial level, and also to make the bioeconomy more resilient. For this, we are on the roadmap at European level, and one of our motto is also MIRI, where the biodiversity meets biotechnology and the bioeconomy, is one of our points. Who make the consortium so far? In bold, that is on your left part of the screen, you find Belgium with different collections, France with different collections, mainly two institutions, Institut Pasteur and the INRA, that is the Institute of National Institute for Agriculture and Environment nowadays, Latvia and Portugal. And you can see that we have several collections here, and we are now in large collections like the Institute Superior Technical collections from the related with Professor Isabel Sá Correia, and other collections they asked to join this infrastructure. We have Spain also as the funders. Portugal is where the headquarters is, and we co-host this infrastructure with Spain, mainly with the University of Valencia, because they are responsible for the informatic part of the infrastructure. The other countries, Greece, Italy, Netherlands, Poland, Romania, are prospective members. They are on the process to make the adhesion and to be members. like Italy nowadays, Chile in South America, and you signed maybe 1 month ago memorandum of understanding with the US CCN, United States Culture Collection Network. We not merge the collection from the United States with what we are doing at European level, but what we decide that there are common points and common studies, like the impact of bioeconomy, of the microorganism in bioeconomy. Now we are trying to make a big study at global level to try to understand why the microorganisms are so important, or if they are so important from the economic point of view. The structure is quite slight so far. We have a central coordinate unit. From the right, we have the executive director, that is Luis, the secretary is Anna, and on the bottom, we have the IT officer, the access officer, that they have changed. And Tony is in Spain, Adriana is in Institute Pasteur, Basen is financial officer, is with me in Braga, the other ones also with me. And we have this structure, it's a slim structure, and we have a European project that pay, and I'm the coordinator of this project with Armand, that is also the co-coordinator. So we are a few people working, and we rely on other people and other institutions. If you go to Braga, you can visit us now in our headquarters, that they were inaugurating 13 December, not 2020, but 2022. I was in different times. We have an agenda, a strategic research innovation agenda, that we call microbial resource for green, health, and sustainable future. And if you look for the agenda, we'll have 7 strategic areas: health and food, research on pathogenic and microorganisms, and human-animal infections disease, the research and development new pharmaceuticals, high potential, but also including antimicrobials, vaccines, phage therapies, and microbiome therapies for human use. Microbiome therapies is quite new, and it's an emergent field. It's quite important. In agro-food, the research and development of new, safe, healthy, and sustainable food and feed products, and the resource and the methods for biological management of soils and crops. And finally, environment and energy, of course, that we are related and work with the pollution and environmental problems, and even with the pathogens that comes from the from the environment, as a concept of One Health, and research and development of renewable bio-based chemicals, materials, and bioenergy source. So this is what we do. And finally, the last but not least, rescue and preserve the microbiome biodiversity. So in this sense, I don't go in detail. You can check this in our page. The most important is that these research 7 strategic areas are very well aligned in health and food, agro-food, and environmental energy. Of course, that our key pillars are biodiversity, biotechnology, bioeconomy, sustainability, and One Health. And don't forget that with this approach, we cover 7 sustainable development goals from 8 from 17. This is quite important, this alignment. And the roles of microorganisms are so important in this sense that we need to take care about this. If you go in more detail, you can cross and make these metrics, you can cross the different expertise that we have. And we have so far in this single catalog more than 400,000 strains. I think that today or in the near future, during this week, we'll deliver more 200,000 plasmids in catalog. It's unique. We don't have any economic bloc, United States or in Asia, that compete with this catalog or catalogs. And we can see that different biological materials, different microorganisms, of course, filamentous fungi and yeasts, don't forget that yeasts are also fungi, can cross very well these different areas. So it's absolutely important to have this concept to understand what we can do and what we can help. How can we help you to make good science? Don't see the collections as a single repositories, like a library with a lot of books, but nobody go there. No, the collections have a high expertise on the microorganisms that they preserve and they put on the catalog, and these expertise are service that can offer to you. And you can contact us and we apply to grants, because we are able to grant you to learn these techniques with us. That comes from the microorganism isolation, preservation and cultivation, screen tests and bioassays, characterization, different characterizations, a different service, taxonomic databases, and consultants training and contract research. Don't forget that times to times we open calls and we pay for the students, for research, for the teams to go through this research infrastructure. And it's paid not only the travel, the accommodation, and everything that we spend on the laboratory. So it's completely free for you. We have these calls open time to times from MURI, but not only MURI, all the research infrastructure are open. I belong to another project that is about cancer. We have now 0.5 million euros open for calls for curiosity-driven research. If you have a curiosity to try to test this, please apply a project. Maybe you get the money to do this. We have a cutting-edge technology that goes from sampling with the different technology. I don't go in details, for isolation, identification, bioresource data, to for the application. So in this, we have the different technologies, the expertise to understand, to training you in different technologies. And if you don't have the technology in your laboratory or in your institute, nowadays, you have a big laboratory that is MURI and is a European scale laboratory. This is the new concept. So move forward and move around the laboratories. I don't go in details. We have a platform. You can consult the platform. Of course, that we have the catalogs, we have the service, we have the training, we have the expert clusters. We are now set up the expert clusters. That means that all the experts that we about one single topic at the European level, we have with us to answer by directly with you, by chat or by in a different way, and you can ask us how to do this, how to solve that. And we have the expert clusters that are available to support you online directly, or if the situation is more complex or more confidential, you can also to address this in a different way. But the cluster experts are now set up also on the platform. The catalog is there. I only play a little bit with this. And to finish, I try to tell you that why the microorganisms is so important. In 1 year, with this MIRI has much more here. We start in 2008 speaking about MIRI. But in the last year that we became a legal entity, we applied for several projects here. The two first projects are related with COVID, and Isidore is related with the important project that tried to provide Europe to be prepared for the next pandemic situation. If you look for the money involved is 21 millions. I'm the board. I'm going to Paris because we have this meeting about Isidore. The first one is related with only informatics, and we try to learn from the different database for COVID. We try to learn what we can extract and make meta-analysis on this. Is a small project, is 12 millions. But MIRI is on board. It's all involved in all this. I'm involved in Cancer Serve because Cancer Serve is related with cancer, but don't forget that some cancers, they have microorganisms, virus involved in the origin of the cancer. But the most important, as it was said before, microorganisms, they produce bioactive compounds that they have very important anti-cancer activities, and we need to explore this huge biodiversity that we have on the collections, and we make bioprospecting, and we continue to isolate from the nature to try to solve the problems of the cancer. And the Cancer Serve is a project with 50 millions. Now is ongoing, and we have a call with 0.5 million explore the curiosity. AgroServe is related with the research in agriculture and ecological transitions for sustainably more resilient agriculture at the European level. We are also involved because the microorganisms are impacting agriculture, of course. We have now the micro project that is how to preserve microbiomes. We are very good in preserving microorganisms, but not so good in preserve the microbiomes. It is new, and it is a challenge. So we have this project. Now we finish negotiations of about 6 million, and the bioindustry 4.0. That is also we are involved in this new project about 10 millions. With other infrastructure related with the new concept of bioindustry and the digitalization, the use of different approach to produce, to make the industry of the future more sustainable, more eco-friendly, and more digital also. And 2 weeks ago, not 2 weeks ago, 10 days ago, we submit a new project that we call, and I'm the coordinator of the project, the proposal, we call Microbiomes for Climate. I hope that is approved. It's about 50 millions of that we ask to discuss the role of the microorganisms and fungi in particularly to make the terrestrial ecosystem more sustainable. This is, again, is a quite complex project. It's huge, and we involve also the United States of America in this project. So we are not working alone. We have a lot of different institutions and other research infrastructure, ERICs. So the different ERICs, we work all together in a top layer of the management of the science at European level to bring the new ideas and bring money to the science. So in this sense, we have another sustainable development goal that is 17, that is the partnership. We need to make partnerships, we need to work all together more and more, bring all the different science, different complementarities, multidisciplinarities, integrating the knowledge to try to find solutions, innovative solutions, and at least to find or to help to face the big challenge that we have nowadays in our society. So I finish and I thank you, and you can visit us on the website and if you go to Braga to the headquarters.
Thank you very much for telling us about this important project and platform that covers all type of known and unknown microorganisms. So now we will have a discussion period. I think there are some questions in app. How can I? Okay. Okay, so the first question, it is for Miguel. Is it possible to produce materials from waste effluents, or do they require very specific and controlled conditions in order to assure the best quality possible?
Okay. Do I answer it from here?
Yes, yes.
Okay. So the whole purpose of growing micromaterials is to use waste streams. So there is no purpose of just having food to grow as or to act as a substrate, or else we are diverging food from people to growing materials. So the idea is to really work on waste, agri-food byproducts, even sometimes just secondary products from some industries. So the idea is to use nothing that can be related to food and can be applied to this kind of circular economy. That's the whole purpose of all these kind of projects and companies. They all intend to do this, and not only because of that, is the fact that if they use materials that were expensive, so the micromaterial will not be sustainable even in an economic point of view. So they have to have cheap materials because growing fungi is expensive enough to a good price to a material.
Okay. Another question that is here is, do you think, is also for Miguel, I think that now we only have questions for Miguel. Do you think that consumers are ready for these new fungi products? What is the bigger challenge to introduce them into the market?
So
Those,
okay.
I think those final.
Okay, we can have two perspectives. So it's the consumer and the producer of of this kind of materials, or the business-to-business client. So one, I will answer by examples. So IKEA in some countries, they sell this, furniture, and in the corners of this packaging, they used to have styrofoam, Portuguese, and so in the corners they use this kind of parts of styrofoam to protect the furniture because of the transport. In these countries, they use the ecovative byproducts or ecovative products to protect the furniture. So it's a reality in some parts of the world. In terms of consumer, not only because of we are all contaminated with the need, but well contaminated with the need that we have to be more greener in our approach, not to be one-time users, not overspenders. So the fact that we have these materials that are needed to packaging or for other kind of process in the industry, if we can have a greener destiny or final destiny to these products, it's not only a point of view or a point that we are just creating a greener destiny, but also we are doing some good to the world. Because the fact that these materials are growing from waste, and then in the end they can be, naturally decomposed. So the client is not the problem. Normally it's the industry that has to change the production process that are more problematic. But as I just showed by IKEA, that is a monster in terms of industrial processing. If they give an example, the other will follow for sure in a short period of time.
Okay, related with that, I have one question, is that about the stability of those type of materials, for instance, for packaging and textiles, how long does they stay in that form without degrading and so on?
Well, of course, I cannot speak for all the companies.
Okay, of course, but in general, in average, more or less.
Yeah, but I can speak for the ones that I presented, Microworks and Ecovative, because
Yeah.
I already received some samples and bought some materials, and I already tested them. Especially the first one, Ecovative, has almost 20 years of industrial processing and testing and R&D. These kind of products are, has the same performance as the, synthetic products, even in packaging, even in the isolation properties. So they mimic very well the well-known materials we have using fungi. The only challenge for now is to down price in order to be more easily sold and easily adapted by industries. So in that point of view, in terms of performance, there's no challenge there. Even Microworks that does this kind of leather is cheaper than leather. Because even if leather comes from byproducts of the slaughterhouse, they spend a lot of money in energy, and nowadays we well know that gas prices, it means energy costs, and they are higher. So using this kind of material that grows at ambient temperature, or instead of using the steam or other kind of more rough process, of course, nowadays they are even cheaper, and in terms of performance, they say that they have the same properties, and at my point of view in testing, I don't see any problem with this kind of materials for the applications they are applying, shoes and clothes.
So it is. They also use natural resources, but it is an alternative, an important alternative to other type of
natural resources, but always thinking about waste streams,
secondary products,
not going for this kind of production.
Okay, I have some another more practical question is how do you inoculate the fungi in the trees out of the lab? Do you use a pre-inoculated soil to plant the trees?
Okay, I can explain. So, as we saw in the first presentation, not mine, from Professor Maria Luz, we grow our fungi in petri dishes the same way. We can do even liquid fermentations as our main way to grow fungi and to have big volumes. And that's the starting point. And then when we bring this kind of fungi to the greenhouse, of course, we have to adapt a little bit the soil that we use to grow the plants, and especially the timing, because the fact that we are connecting fungi to roots, we have to wait until we have some roots and some specific type of roots that are appearing. When we know that they are, well, we apply them. Apply them is just put some mycelium over the roots, having these conditions by temperature and humidity, and especially pH, to improve the connection between, or in this case, nowadays we can say that is an infection, a beneficial infection from the mycelium to the root, and then if it is arborescent, gets in the root. If it ecto, it covers the root. But the main mechanical process is just this, and even the fact that ectomycorrhizal especially are very easily grown. It takes time, but they are easily grown in the laboratory. Even the companies that sell mycorrhizal plants, they produce the mycelium in lab, not in big tanks or nothing industrial, just lab scale.
Okay, thank you. Now I have two questions that I am going to join for Professor Nelson Lima. Is MIRI currently involved in projects with industrial partners, and what prerequisites do you need to order a microorganism from MIRI?
Okay. Yes. One of the priority of MIRI is not only bring science to scientists, but bring science and facilitate the access of biological resource and the data associated. Don't forget that most of the time the microorganism itself is important, but more than this is the associated data that we have, and you can extract directly from the catalogs or indirectly if you contact the collection, because the collections normally they know much more than 5 or 7 minimal data set that we put on the catalog. We have a big database with a lot of information that is not big. So the communication should be in this direction. And of course that the bio industry for us is quite important. Is the reason because the collections, most of the collections are, they have the important partnership with the companies, some of them big companies. At MIRI level, we are working more at political side and with different stakeholders, but of course that each collection keep their partnership, and we try only to put more visibility on this flag. We rely on the work on each single partner in this sense. The other point is the price, how to get. Yes, nowadays if you find on the single catalog of the MIRI, if you find the strains of Mycoteca, you have or another collection from the Institute Pasteur. You have two possibilities, or you go direct to the collection, because all the collections are open and public service collections, so you can order and make the negotiation, or go to the MIRI. Go to the MIRI, you will be charged by MIRI to deliver the strain. However, you can also get some strains free of charge if you can apply for if you have a call to deliver strains free of charge, and we have if you get the right window that is open to get the strains free of charge, you can get this free of charge from MIRI side. If you go for MUM or other collections individually, no way, you must pay the price that are on the catalog. but is some way how to deal with to work on the different layers of the structure. But we try to improve this, and more and more the discussion at governmental level is that the fact that through MURI, each country, each government pay the strengths. But it's a negotiation of the governments, so it's always tricky. But we are on this on this way to make this more open and free and not cost too much to get strengths.
I have one question related with that. I'm not sure if I understood well. Because you spoke about the calls for curiosity research projects, but that is to obtain the strains or to get opportunity to work in the...
You can make it's quite complex structure, but if you have an idea, you can go to different service, or you can go also, you can propose a project to MURI or to other research infrastructure with a workflow, saying, I need to go to Braga to get these strands, I need to go to Paris or to another part to do this characterization, I need to jump to another research infrastructure that is bioimaging to get confocal analysis, or bioinstruct to make the characterization of biomolecules. So you can organize a project of this, not with a single author or a team with a PI and 2 or 3 PhD students, and you can apply. If you get money, you can move through these paths, not in a single site, multi-site, not in one time, in several times, not one single person, but in team. Yes, it is possible. What is the most important for us? We rely on the international panel of evaluators, and the major point for us is if the project has, we found the project.
Normally, who applies? At the PhD level, PhD students, or
PhD student to senior scientists.
Okay, not for graduation students.
Students normally first year is quite difficult because it's not consolidated, unless to have a tiny project. That all these projects that we found will never be co-authors. The results belongs to the author of the authors of the project. We never be involved in co-autory of papers. Only what we demand is a knowledge of the grant and of the research infrastructure. We never be involved as authors. So this is an important interest to be authors or to be co-authors on this sense. It's not our purpose.
Yes. Even if it is not for you now, maybe it can be in the future. So you should keep this information. So another question is related with microremediation. Can it be used? Yes, I think so. To remove recalcitrant pollutants from the environment or from wastewaters, because there is another question related with wastewater treatment. Using fungi.
Yeah, many examples are in terms of our lab. We work more in terms of soil, remediation of soil, but already saw some articles about marine and water remediation. So yeah.
Yes, yes. In my laboratory, we use this for many years to remove textile dyes or other recalcitrant organic compounds. So fungi are quite good in this sense, mainly because they have some enzymes that Professor Maria da Luz spoke about, some not hydrolytic enzymes, but more oxidase enzymes that are involved, because are not so specific in this sense, and they can help us in to get a good performance in biodegradation and bioremediation in this sense, even in wastewater treatment plants, and there are some cases that are in the pilot scale. I have one question for Maria da Luz, if it is possible, because she told us, and for you it's quite important to understand why she uses ITS as a barcode for identification, and she clearly said that she is happy with ITS to get the identification at the genus level. However, if she needs to go to a species level, she changed for a different barcodes and different genes. Can you, I don't know if she's around, can you explain a little bit, because we have students here and normally what we learn in most of the time that is the ITS is the universal barcode for fungi, for identification, but in fact, and in her case, that she works mainly with aspergillus and penicillium, I'm sure that ITS is not enough to go to the species level. So can you elaborate a little bit more, please? And what the alternative genes for to go to the species level?
Okay. Because I'm not.
It could, maybe she didn't hearing me well.
Well, that's okay. I'm not seeing you, but I can answer that. So
Okay.
in some cases, yes. In most cases, particularly when with aspergillus and penicillium genus, it was not possible with ITS to go to the identification. So in those cases, we used it. I think it was beta-tubulin and camudula genes to get the identification to this.
Okay.
We in some cases we use it also RPV2, and I think another region. It depends of the group.
Yes.
So
yes, it's only to make sure that the audience stay clear in this aspect, that ITS is good to start, but is not good enough to finish.
Yes. No,
is the message.
not sufficient resolution to find out the species within a gene. So I have another question from the students to Professor Maria da Luz Calado, that is, have you ever discovered any potential application of a particular marine fungi by accident? By accident.
By chance.
By chance. By chance. You are not looking for it and you got it. Something interesting.
I'm not sure if I understood the question.
Maybe if you find a fungi, but you isolated the fungi, but you didn't. You are looking for some type of activities, and in the end you find some other more different one, different than that the one you are looking for.
Okay, so at the moment, no, but I'm expecting to have that surprise. I'm hoping, because I'm very happy with my collection, and Well, the thing is, we are trying. Well, we did the fermentation assay with that medium, with those conditions. But we know that if we change the conditions, the culture conditions, probably the fungus, each isolate will produce a different compound. So we have so many things to check and to change, and probably, but we will have to focus. And at the moment, we have so many isolates
Yeah, I
to check.
understand.
So we are trying to not to spread too much. But so far, we didn't find anything by accident. But we never know.
Okay, I have two small questions related for Professor Maria da Luz. One is, I noticed that you use sea foam. I suppose that sea foam, in order to isolate fungi, and sea foam is that white material that accumulates in the beaches, for instance, after storms, that sometimes we thought that it was pollution or something like that, but it is not, because what do you...
Sometimes they carry the sea foam, they carry spores and also propagules, the vegetative propagules, so we can isolate the fungus that come from the sea, from that foam.
Okay, it's very interesting, because some weeks ago I also read something about having microalgae and interesting cyanobacteria and microalgae in those white foam that sometimes we think that it is not important, but it is a rich material. The other question is related with I think I understood that you of course these works are dependent on the cultivation of fungi, but there is some limitations related with that are related with the non-culturable microorganisms. What is and also for the students to because it is something that we speak in the microbiology course and so on. What is, or do you have any number for the percentage of fungi mostly, that are not culturable, that you can be missing, because sometimes it's difficult to cultivate, to isolate this type of microorganisms?
At the moment, I don't know, but that student that I told you before, that he's doing his thesis his focus is thesis in the Nicholas fungi, is going to do also probably a metagenomic analysis, and probably crossing the data, we will have an idea if we are losing.
Yes, because I don't know. As well, also for fungi, we speak about some numbers for the whole microorganisms in environmental samples, but we don't know what about.
Yes, when I isolate, I didn't say, but I use two different media.
Two different,
one poorly rich with nutrients and another rich, to get different things from.
Just to try to overcome, or at least partially overcome, this type of problems, because this is an important limitation. Okay, so
only to underline.
Okay.
Because nowadays when you cultivate, use different media to get better chance to isolate the cultural microorganism and not viable but not cultural, we call cultural mix. And nowadays cultural mix is important area that is necessary to combine with metagenomics. So nowadays metagenomics only is not enough, or dependent culture is not enough. We need also metagenomic plus cultural mix. So be aware about this buzzword. That is an important concept that we need to develop also.
Okay, so microbiology is an ancient
based on
area, but is
moving forward.
We are always learning and doing new things and try to get more information. So do someone wants to ask some more questions, or can we finish? Maybe we are a bit out of time. We can finish this section. So thank you very much.
Thank you.
And
Just to let you know, the winner of the giveaway is Rodrigo Nunes, so you can come here. And thank you all for being here today. Thank you, our moderator and our speakers and our speaker here. Thank you.
Thank you.
Obrigada outra vez.